Thank you. Good morning, good afternoon, and good evening. I'm Kathy Orsted from Gilead Sciences, and I'll be the moderator of this webinar. It's my pleasure to introduce Dr. Monica Burns and Dr. Sim Salian Mehta. Sim is a board-certified senior project toxicologist at Gilead Sciences for the past 3 years. She is a non-clinical project lead responsible for the strategic and scientific development of a broad range of modalities, with a primary focus on oncology, inflammation, and virology. Sim has With that, I'll turn it over to Monica and Sim.
Thanks, Kathy. Good morning, everyone, and good afternoon, wherever you are. We are today leading of the series of our 3-hour webinar series, and the first of our talks will be on the cross-industry husbandry survey findings and its impact on NHP health issues. Monica and I will be sharing this talk. A part of it will be covered by me, followed by Monica giving you the entire details about the survey.
while they were closely connected to the in vivo work being done as scientists at the pharmaceutical company or others very heavily involved, they were not actually at the CRO itself, seeing these studies being conducted in person in most cases. So we did get a lot of responses of don't know or rely on CRO practices. This, of course, goes hand in hand with a lack of CRO perspective. That's certainly something missing from this survey and would be a good area to focus on for any future work. We also had a limited sample size with an uneven distribution of NHP source and study location. So there's a bit of loss of normality there. Ultimately, what the goal of this survey was to conduct benchmarking. So getting feedback across the industry. What are we seeing? What are the trends? What are we doing about these trends? And we wanted to use these responses to create some recommendations for best practices to minimize NHP health issues, specifically focusing on this pre-study period. Next slide, please. So the first section of survey questions addressed demographics and impact of NHP origin and source on NHP health. Most of the respondents conducted studies at North American CROs or in-house, and they used, for the most part, Chinese or Cambodian origin NHPs. You can see that data reflected in the middle top graph. 14 of 33 respondents reported having an impactful health issue, such as abnormal feces or respiratory findings, on more than one study, with the highest incidence of these issues observed in Vietnamese origin or sourced NHPs. You can see the sourcing data covered on the middle graph that is at the bottom. Then 11 out of 32 respondents indicated that the NHP origin or source was not a causative factor for increased frequency of abnormal feces. So we do have some mixed messaging about whether sourcing is impactful or not. We do know, and overall, I would say the body of literature related to these differences for different NHP sources is a bit scarce. We do have some publications that address things like differences in GI microbiota across different sources. We also have some publications that reflect an increased lymphoplasmacytic cell infiltrate in the GI tract of Asian origin NHPs. We also know that Mauritian origin NHPs typically do not carry some of those simian viruses that we are most concerned about. However, there's still a lot of work that needs to be done to really clarify the differences between NHPs that are sourced from these different geographies. And again, here, this is maybe a theme that you will pick up throughout the discussion here. There is a need for a CRO perspective and a more focused survey to really define what the differences are in NHPs sourced from these different geographies. I'll also point out before moving to the next slide that throughout the presentation, you will see the graphs. The blue includes respondents without health issues, and the orange includes respondents with health issues. So you can carry that through the presentation. Okay, you move to the next slide, please. All right, so then the next, after looking at demographics, we asked our survey audience about the impact of secondary quarantine practices and refinement practices on NHP health. So here we talked about the acclimation period, and this is acclimation / secondary quarantine. So we are talking here about animal movement from the supplier, from the domestic supplier to test facilities. These periods are typically, there is variability in these periods depending on where the animals are ultimately moved to. These periods can be impacted by the age, the perceived stress, and then what is being evaluated later on in the study. 17 of 29 respondents indicated that they used an 8 to 29 days of acclimation as that secondary quarantine. So again, arriving from the supplier onto the CRO site. Typically, it is thought that extended periods of quarantine, of secondary quarantine, so more than 15 days, will increase monitorability and detection of health issues. You'll notice from this top graph on the right that we had about relatively kind of similar rate of detection of health issues in that 8 to 14 days and the 15 to 19 day period. I will note that at least a few weeks of quarantine or acclimation is typically required by facilities for best biosecurity practices. We also found that preventative and pre-study health practices like dietary modification, environmental enrichment, and behavioral assessment training had the biggest impact on NHP health. And I'll highlight the behavioral assessment because that was correlated with lower health issues, presumably due to lower stress and increased monitoring. With those behavioral observations comes the ability, if you're looking at the animal more, you would notice more going on with them in terms of their health and behavior. At the graph on the lower right, you again see, and it's a little bit small there, but if you're interested, please pull up the paper and take a closer look at the graph. But you'll again see that we have the orange, that's the responses that are associated with animals having health issues later in the pre-study period. And then the blue are those that are, in this case, using refinements without health issues. So that's just one to highlight. And anytime you see a difference between those two, a big difference between those two, those are areas where we have focused on, we have pulled out and focused on as recommendations, kind of as a broader takeaway from this benchmarking survey. Okay, we can move to the next slide. After looking at our secondary quarantine / acclimation period, we looked at the pre-study exclusion and impact of NHP health. We found, perhaps not surprisingly to this group here, to these attendees, that the most common health problems leading to pre-study exclusion were abnormal feces, with 17 out of 33 respondents indicating that was an exclusion criteria, and then also wasting and failure to maintain body weight with 17 out of 33 respondents. We also found that stress-related behavior, so for example, something along the lines of alopecia or other abnormal behaviors, were common reasons for exclusion, and then ECG and ocular abnormalities as well. So again, with this graph, we have our same pattern. Blue does not exclude with health issues, and then orange does exclude with health issues. So we have anywhere we're having a bigger degree of separation between them, that's where we'd encourage people to focus on and make sure that you would encourage folks to think about excluding for those purposes as reported by survey respondents. We also found that the implementation of pre-study exclusion criteria, especially those focused on general health, like body condition and pathogen screening, and stress behaviors like severe alopecia, resulted in fewer health issues. So I think for those that are heavily involved in this work, these all kind of make sense and align with what we typically expect. So it's useful to see them benchmark across the industry that these are the criteria that we feel most strongly about excluding animals. And in that way, when you take those animals out pre-study, you're less likely to have health issues later on still in the pre-study period, but later on. All right, thank you. Next slide, please. Then we also benchmarked serology and fecal testing practices. We found that 60% of our respondents relied on a classic macaque panel for exclusion criteria. And by macaque panel, we really mean that herpes B, SIV, SRV, and STLV. Over on the left, you can see that serology data reflected. So useful to know that that core macaque panel really, the majority of facilities and reflected in the survey responses are using that for exclusion criteria, using those results for exclusion criteria. Our survey results suggested that there is a consistent NHP serology testing and pre-screening practice that is kind of generally used across the industry. So again, that's reassuring that for the most part, across the industry, we as the stakeholders kind of looking at NHP health pre-study are using in large part that same criteria. However, we'll talk about the frequency of serology testing as an area where we'd make a recommendation. Serology testing is typically conducted at least once pre-study and pre-shipment. That was reported by 11 out of 31 respondents, with some conducting serology screening more than once. So by some, I mean 3 out of 31, so not too many. Many respondents here did not know or deferred to the CRO. So this is that main limitation here where There is close connection to this work, but not typically a direct on-site connection, except for the in-house studies. For those respondents who performed serology only once, 64% experienced health issues versus 25% who do repeated screening. So this is kind of the main takeaway of this slide here, I would say, is that increased viral screening is recommended. So yeah, if you're screening just once based on our survey data, you would be then more likely to experience health issues later in that pre-study phase, versus if you're screening twice and excluding appropriately, you would be less likely to experience NHP health issues in the pre-study phase. Next, I'll move on to fecal testing. So over on the right, you'll see a table that represents the fecal testing response data. Here the respondents were asked to respond about testing when we have animals with health issues. So this is not all animals here, just animals with health issues. It is conducted prior to placing NHPs on studies. For many of our respondents, few did not conduct any fecal testing. Few also only do it when there's abnormal feces. And then again, a large number don't know or defer to the CRO. Then I'll jump down to the final bullet point, that while fecal testing is routinely done prior to animal arrival at the facility, reported by 15 out of 19 respondents here, increased fecal screening prior to placement on studies is recommended by this working group to minimize potential health concerns. So here I'll just highlight, we have kind of consistent NHP serology practices across the industry. We'd recommend increased viral screening and increased fecal screening prior to placement on study. We can go to the next slide. Then we'll talk about abnormal fecal observations. So again, perhaps not a huge surprise to those of us who work closely with these animals and with this type of study. 22 out of 31 respondents indicated that persistent or unresolving abnormal fecal observations is a justification for animal exclusion. The recurrence of fecal abnormalities were higher in respondents who did not necessarily exclude animals with abnormal fecal observations. So if you're seeing abnormal feces, you are, according to our survey data here, perhaps likely to see that occur again. It was unclear from our survey data kind of the impact of age. So I think one of the questions that the survey team had come up with through our experiences in recent years was, are we seeing younger animals enrolled on study? Are we then seeing more abnormal feces related or kind of directly correlated to that inclusion of younger animals? And based on our survey responses, we didn't see that. We didn't see the answer to that question being yes. We saw it as not sure or perhaps no impact. We saw 19 out of 29 respondents that frequently saw body weight loss and dehydration correlated with these abnormal feces. So not just, diarrhea with nothing else happening. We are seeing other impacts as well. And then fewer respondents saw no correlation there. This again, I think is not surprising data, but good benchmarking to have overall. The majority of respondents requested veterinary consultation for abnormal feces. And here I will point over to the graph on the right. So most respondents are going to call for veterinary consultation when we have abnormal feces that are observed for greater than 3 days or 3+ days, 3 times a week, or after a single incidence with blood and or abnormal behavior. Here we do still see that don't know or defer to CRO reflected. However, there is a lot of agreement across the board from respondents when we are calling in the veterinarians for observation and treatment. Okay, we can go to the next slide, please. Then continuing on with the theme of fecal abnormalities and working up those abnormalities, we will further discuss the diagnostic tests that are used when we see abnormal feces and animal health impact. So here we saw that a decline in NHP health, or survey respondents reported that when they saw a decline in NHP health, they would be more likely to use diagnostic testing. So not just testing everybody, but that correlated with the NHP overall clinical health picture. We found that 10 of 33 respondents reported unfamiliarity with diagnostic testing or deferred to the CRO. So here we do, again, see a significant percentage of respondents showing that gap between the study teams and the CRO that is conducting the in vivo work itself. So when an animal is showing compromised health and diarrhea, these respondents would defer to the CRO, or they're not sure what diagnostic testing is being done. The rest of the respondents are aware of the tests that are done and or are involved in making decisions about diagnostic testing with the CRO partners, but we do still see, about a third of those respondents not having heavy involvement in that testing or decision making. This again is one of the perceived gaps identified in the survey and very much highlights why the CRO perspective is needed here. 29% of respondents overall found that fecal testing was useful, and interestingly, only 32% found that histology was useful in understanding the underlying cause of abnormal feces. Our respondents felt that a combination testing paradigm might be better than in general evaluating in isolation. And it's possible that respondents may have defaulted to not helpful when diagnostics were only useful in a small percentage of cases. So this finding with kind of a lower rate of histology being useful than expected could have been related to the wording of the survey itself. And that's another limitation and is kind of a watch out for survey questions overall. The questions have to be formulated in the exact correct manner to get the useful data. So luckily we only had a few cases. This is probably the biggest one throughout the survey where we felt like that occurred. On the right, you'll see a table that represents kind of different diagnostic tests on the left of the table, and then also shows data of respondents using it for otherwise healthy NHPs and then NHPs that have a health impact. I'll point out serum chemistry and PCR as two tests where we see a much higher rate of respondents using those tests when NHPs have health impacted versus when they are otherwise healthy. And certainly necropsy and histopathology too, but not possible, usually not possible in otherwise healthy NHPs unless you're doing some sort of biopsy. 20 of 28 respondents did not change husbandry practices versus 8 of them who either quarantined, separated animals, conducted additional diagnostics, or provided treatment to all animals in room. So this shows that we're doing diagnostic testing and we may have some individual treatment of the animals, but overall husbandry practices are really not altered by the results of those diagnostic tests. All right, now I'll move on to the next slide, please. Now we'll talk a little bit more, again, about abnormal feces. You'll notice a theme throughout this presentation. Here we'll cover the survey questions and responses related to veterinary treatment and use of environmental enrichment for NHPs with abnormal feces. So the most common treatments for NHPs that were otherwise healthy but presented with abnormal feces were dietary modifications like a bland diet or an increased fiber or liquid diets, and then also probiotics followed by oral electrolytes. So we see that kind of given to all animals in general, not necessarily just the healthy or unhealthy animals. However, for those animals that had a decline in their health, that had just their overall clinical picture deteriorating or poor, there was an increase in survey respondent use of antibiotics and subcutaneous fluids as frontline therapy, and then other therapies as well. But we saw this quite consistently across the board with our respondents, that when we see deterioration, we're moving on to that first line of real treatment versus just the supportive care with dietary modifications and probiotics. 61% of the survey respondents suggested that sponsor involvement there was sponsor involvement in treatment decisions. So that reflected that a majority of survey takers had been involved in those discussions when it came to kind of veterinary workup, care, and treatment of NHPs in the pre-study, NHPs with abnormal feces in the pre-study phase. And I will just point over to the graph on the right again. So the orange is abnormal feces with compromised health, so not healthy. And then blue is the persistent abnormal feces, but otherwise healthy. And you'll see those, the area where you see the most kind of disagreement in the bar graphs are where we see different treatments selected for those animals that have poor health. Okay, we can move to the next slide, please. And then here, and this is a section that we expand on quite a bit in the paper as well, but we noticed some pathogen trends and trends for managing non-human primates with fecal issues. The most common enteric microbiological agents screened for included Shigella species, Salmonella species, and Yersinia species, with few respondents testing on a regular basis for Campylobacter species. For the Shigella, Salmonella, and Yersinia in particular, sometimes survey respondents indicated that they felt that these organisms could be present on a more subclinical basis, but they could lead to disease, recrudescence of disease, and stress or immunocompromised animals. We also have discussion in the paper about the challenges of kind of isolating and confirming these organisms. So there are limitations of laboratory testing for sensitivity in terms of sensitivity and specificity here. There's also a higher prevalence of antibiotic resistance in some of these organisms. And then I will also point to, there's a line in the paper that discusses that some, while fecal culture is done in many cases, sensitivity is not always selected along with that culture. And so that's an area where we would encourage a sensitivity panel always to be run with a culture for these potentially pathogenic fecal organisms. There should be kind of a discussion and alignment between where the animal is coming from and then the ultimate point of arrival about the pathogen status to make sure that repeated treatment is avoided. We all need to keep good antimicrobial stewardship in mind, and if we are constantly treating with antibiotics, that is not helping in that effort. We also do see, especially with fluoroquinolones, it is quite easy to develop resistance. So that's just really something I encourage everyone to keep in mind to try to minimize those just kind of preventative use of antibiotics everywhere. And then also, depending on where the animals are coming from and going to, making sure to avoid mixing of carrier and non-carrier animals, because you might have a healthy animal that is pathogen-free, and then they are placed in a room with a carrier animal. You can have some transfer of pathogen status going on there. Okay, we'll go to the next slide. So you had a number of high-level takeaways from these surveys, a number of high-level takeaways from this survey, even though in spite of the limitations that we acknowledge that the survey had. We found that 42% of survey respondents reported health issues on either 1 to 4 or greater than 4 non-clinical studies. So clearly this is a prevalent issue across the industry if we have such a high percentage of survey takers reporting this as an issue. We also found that our survey data suggested that fecal issues and/or failure to maintain body weight are the primary causes of NHP abnormal health observations. We found that pre-study screening practices like excluding NHPs from study for GI-related health issues, whether they're confirmed potential enteric pathogens or diarrhea and signs of stress behavior, such as alopecia or overgrooming, will result in a study cohort with better overall NHP colony health. Veterinary intervention for NHP fecal issues typically across the industry is either at the point of having abnormal feces for 3 or more consecutive days, or after a single occurrence if it's associated with blood and feces and or a decline in NHP overall health. And then fecal testing and screening practices did vary among respondents. However, there's kind of a core baseline that most respondents agreed on. And further evaluations are needed to determine the impact on NHP selection for studies. The most common diagnostic testing done in the pre-study phase is fecal flotation, fecal culture with or without sensitivity, which should always typically include sensitivity, and then serum chemistry. Less than half of survey respondents tracked the health screening outcomes, and that limits the ability to appreciate evolving trends in NHP health. That is a rich source of data that really represents an opportunity to learn more about overall NHP health. So that's a great area if you're looking to better be able to monitor and manage NHP colonies. Being able to track and mine that data is a good place to do it. And then, of course, we really do need that CRO perspective in the future for this type of work. We found that preventative health practices, especially dietary modification, environmental enrichment, and behavior assessments, including training, appear to have a positive impact on NHP health and study outcomes. This overall is a type of refinement and supports the 3 Rs. In terms of veterinary care, for first-line treatment for animals that have health issues, subcutaneous fluids and antibiotics were found to be the most common treatments. So then I'll move on to the next slide, which is our last slide. I'd like to acknowledge all the co-authors who worked hard on this. This was a multi-year effort, represented a lot of work from a lot of different people. Thank you to Sim again for her leadership here. Thanks to Kathy for her involvement as well. And great effort by the team overall. Thanks to all the survey respondents. It was no simple effort to fill out the survey and to make sure that the data was accurate to the best of everybody's understanding. And hopefully this represents a accurate benchmarking effort across the industry when it comes to NHP health and husbandry practices. So thank you all, and thanks to the attendees today. Looking forward to having some more discussion and questions as we go on. Thank you.
So we do have a question online. lost them, sorry.
Yeah.
And thank our speakers and moderators.